Namaste students, I am Jayant Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth University, Jaipur. Today I am going to discuss about the topic nitrogen assimilation. So what is the nitrogen assimilation? We know that a microorganism has the capacity to utilize the environmental nitrogen and convert it into the usable form. And this is the nitrogen cycle. Similar thing is that uh, the nitrogen which is uptaken by the plants is used for the synthesis of organic nitrogen molecules like amino acids, proteins. These are the synthesized from this uh, nitrogen which is available inside the plants. So what is actually nitrogen assimilation? From this nitrate NO3, nitrate is converted and ammonium is converted, and from this ammonium amino acids are converted. That is nitrogen assimilation. Okay. And it requires large input of energy. It forms some toxic intermediates and it is mediated by specialized enzymes that are closely regulated. Plants assimilate most of the nitrate absorbed by their roots into organic nitrogen compounds. The first step of this process is the reduction of nitrate to nitrate. Okay. First step is reduction of nitrate into nitrite in the cytosol by the enzyme nitrate reductase. Okay. So first NO3, okay, NO3 will be converted. NO3 will be converted into NO2 by the enzyme nitrate reductase. Okay, and FAD and NADH will involve in this reaction. The MOCO, the enzyme that is nominated, and MOCO is responsible for this reaction. Okay, NADPH induces NADH or NADPH. Okay, the most common form of nitrate reductase use only NADH as the electron donor. The nitrate reductase of high plants are composed of two identical segments, each containing three prostatic groups. First one is FAD. Okay. The first one is FAD, second one is heme group, and the third one is the molybdenum group. Okay. So it is composed of three prostatic group, FAD, heme, and molybdenum. And in the molybdenum, the organic molecule is called as the tannin. Nitrate reductase is the main molybdenum containing protein in vegetable tissues, and nitrate levels, light intensity, and the concentration of the carbohydrate all influence all these influence the activity of nitrate reductase, and all the transcription of the translation levels. So these are influenced by these nitrate reductase. These factors estimate protein phosphate that dephosphorylates several serine residues on the nitrate reductase protein, thereby they activate the enzyme. This phosphorylation and dephosphorylation cycle provides more rapid control over this enzyme than degradation synthesis of new enzyme would achieve in few hours. So first nitrate reductase converts nitrite to ammonia. First step was the nitrate was being converted into the nitrite, and now the nitrite is being converted into the ammonia. Ammonium. Okay. So nitrite NO2 is highly reactive and toxic, and plant cell immediately transport the nitrate generated by the nitrate cell from the cytosol into the chloroplast in leaves and plants in roots. Okay, to transport this nitrate and nitrate reductase and enzyme is involved in transferring of this molecule. In the light reaction, the ferredoxin is in reduced form, and after that it comes in the oxidized form, and Fe4S4 come and take the electron and becomes a heme molecule. Okay. The chloroplast and rootplast it contain different forms of enzyme, but both forms consist of a specialized heme group. The heme does redox the reaction and electron flow, and it is just like the reaction of the chlorophyll. And plant assimilate nitrate in both roots. And so how? In many plants, when the root receives small amount of the nitrate is reduced primarily in the roots. If the nitrate supply increases, the greater proportion of the absorbed nitrate is translocated to the root and assimilated there. Generally, species native to temperate rely more heavily on nitrate assimilation by roots than those species of tropical, subtropical regions. Now, talk about the ammonium also assimilation. Plant cells avoid ammonium toxicity by rapid converting the ammonium generated from nitrate assimilation or photorespiration into ammonium amino acids. And this requires the action of two enzymes, glutamine synthetase and glutamate synthase. Glutamine synthetase combine ammonium with glutamate to form glutamine, and glutamine synthesis is stimulated by elevated level of glutamine synthase. Uh, it the glutamate synthase transfer the amino group of glutamine to an intermediate yielding two molecules of glutamate. Now talk about the transamination reaction in which transfer of nitrogen is conducted. Once assimilating into the glutamine and glutamate, nitrogen is incorporated into other amino acid via transamination reaction. Enzyme which involved in each reactions are known as the aminotransferase enzymes. Okay. For example, the aspartate aminotransferase is an example of transfer the group of glutamate is transferred to the carboxyl atom of the aspartate. I'm taking the example of the aspartate aminotransferase. So the amino group of the glutamate is transferred to the carboxyl group of the aspartate, and aspartate is the amino acid which settles reducing agent from the mitochondrion and the chloroplast into the cytosol and in the transport of the carbon from mesophyll to bundle sheet of the C4 carbon fixation. All this require vitamin B6 to act as a cofactor. By biological nitrogen fixation for atmospheric nitrogen into ammonium represents the key entry point of molecular. the biogeochemical cycle of nitrogen. Free living and symbiotic bacteria are responsible for the converting of atmospheric nitrogen into ammonia, and most of these are free living in the soil. A few form of symbiotic association with higher plants, and most of the association plants and microorganisms is between the member of the plant family of Leguminaceae and bacteria of the genre Rhizobium. Nitrogen fixation requires some anaerobic condition as nitrogen oxygen irreversibly inactivates the nitrogenase enzyme involved in the nitrogen fixation. Nitrogen fixation must be under anaerobic condition, and therefore each of the nitrogen fixing organism either function under the natural anaerobic condition or can create an internal anaerobic environment. In cyanobacteria, anaerobic conditions are created in specialized cell called microcysts. Cyanobacteria can fix nitrogen under anaerobic conditions, such as those that occur in the platelet cells. Uh, Symbiotic relations and nitrogen fixation occurs in specialized structures, and symbiotic nitrogen fixation in proteins dwell within nodules, root nodules. And grasses can also help symbiotic relationship with nitrogen fixing organisms, but these associations do not lead to formation of root nodules. So root nodules are the specialized structures in which microorganisms can survive, and they can have anaerobic conditions, and these specialized structures provide a particular environment. And the plant genes which are specific for the formation of these nodules are known as the node genes. And rhizobial genes that participate in nodule formation are called nodulation genes. Common genes are NODA, NODB, NODC. Most specific genes are NODP, NODQ, NODH, NODE. So these all are the genes. So what are the different phytohormones? First, there is infection and nodule organogenesis. Rhizobia attach to the root hairs and release node factor that produce pronounced curling of the root hair cells. And rhizobia get caught by the curl and degrade the root hair cell wall, allow the bacterial cell direct access to the outer cell surface of the plasma. Then infection thread forms, which is formed from Golgi depositing material in the tip of the side of the infection. An infection thread reaches to the end of the cell, and thread plasma membrane fuse with the plasma membrane of hair follicle cell. Then rhizobia release into the apoplast and enter the middle lamella, and this lead to the formation of new infection thread. Infection thread expand the branches until they reach target cells. So this is the basic mechanism by which the microorganism can come in contact with the plant nodules and how they perform nitrogen fixation. So how nitrogenase enzyme complex fixes nitrogen. Biological nitrogen fixation produce ammonia NH3 from the molecular nitrogen. So nitrogen plus 8 electron plus 8 proton and 16 ATP molecule will come, and they will form ammonium. will prepare ammonium and 16 ADP molecule, and they will release 16 pyrophosphate. The reduction of nitrogen to NH3 is 6 electron transfer and is coupled with the reduction of 2 proton to evolve hydrogen. And the reaction is catalyzed by the nitrogenase enzyme complex. Can be separated into two components, Fe protein and MOFE protein. Ferredoxin reduces the Fe protein, and the Fe protein reduces the MOFE protein. And the MOFE protein reduces the nitrogen. So this MOFE protein can reduce many substances, like it can reduce N2 into the NH3, it can reduce N2 into the nitrogen in water, it can reduce N3 minus into nitrogen, N2 plus NH3, it can also reduce C2H2 into C2H4, and it can also reduce H2H plus into H2, and it can also reduce ATP into the ADP plus pyrophosphate in different activities. So my lecture is over here. If you have any query, you can ask me. So thank you very much.